Oh, I know what that is. Okay, sorry for being a little late. I had to reload my browser because the website, I think it was the earlier one, and it wouldn't let me start the lab. So I had to reload it and get that working. Are there any questions before we begin? All right, let's briefly talk about the lecture, not the lecture, the syllabus. And I think that's what I want. Yep. So today we're going to cover Lab 00, lab safety training. I should tell you that for next week, you should read the Infectious Disease Research Project. It's in a file called Project Instructions on Canvas for next week. So let me see if I can find that. See, I needed to reload that page. It wasn't working. The Infectious Disease Research Project. You don't need to read that early this week, but you do need to have it read by next week. The Infectious Disease Research Project. I'll talk about the Infectious Disease Research Project in week 2. All right, any questions about Lab 00? The lab grade is the same as I've talked before in the class. It's just that some of these will be lab grades and others lecture grade. The lab assignments, 5 points each, excuse me, about 10 worksheets. It includes the unknown notes as well, which is worth 5 points. And it counts lab 00 and lab 01 as 1 for 5 points, meaning they're each 2.5 points. Let's see, identification of the unknown, that's a lab project. The infectious disease project is also a lab project. But the grades total to 720 points. Safety information for students and physicians. The following information is only included for educational purposes. As future allied health sciences people, it's important to be aware of these issues. For an online class, though, you will not become infected by a microorganism because everything we do will be done online. But it is important to read and understand about this. In the lab, assuming we were to go into the biology lab, we would have the lab operating at biosafety level 2 or BSL 2. And that means we have personal protective equipment. Students are required to wear a lab coat. They wear gloves. If they're working with a splash hazard, they have to wear goggles. Access to the laboratory restricted. And we work with infectious agents. Now, most of the organisms we work with are not biosafety level 2. Most of the organisms we work with are biosafety level 1, but still we operate the lab under biosafety level 2 conditions. I'll let you read about students who are immunocompromised or working or living with people who are immunocompromised, because obviously we're online, so this is not a concern. Sorry, I gotta blow my nose here. I'm trying to stay out of the picture. I don't know if I'm out. This is a species list of what organisms are found in the microbiology lab. Not all species will be found every term, but these will be the species found, I don't know, all the time or some of the time. Biosafety level 1 organisms, and then biosafety level 2 organisms. Laboratory rules and safety rules. We're not really going to go into the lab, so you could say you don't need to practice these, but you do need to understand them and why we have them in place. Because you're taking microbiology, you do need to know the rules of working in a microbiology lab. We're not going to physically meet in the campus, but learning the lab safety protocols is required. And many nursing programs report that safety is one of the main skills that they want their students to learn from microbiology. Safety is of primary importance in the microbiology lab. The attitudes and actions of persons working in the lab determine the level of safety. So therefore, develop a positive and respectful attitude towards microorganisms. Never take shortcuts or risk the safety and accuracy of the lab. Normally, you're not allowed to take any personal items to your lab bench. They must remain in your book bag or your coat. And those are kept in a cubbyhole, which is not considered a part of the lab. So the very door of the microbiology lab, and then the space from the door to the cubbyhole is right next to the door, is considered outside of the lab. So you walk in the door, and then you put your things in the cubbyhole, and then you walk to the sink to wash your hands. Walking to the sink and any place else you might walk to in the lab is considered the lab proper. Any questions about any of that? Anything that is brought into the lab proper must remain in the lab and cannot leave the lab until it has been disinfected or autoclaved. Autoclaving will remove the organisms from it. And so if you brought like a paper or a pen and worked with it in the lab, you cannot take it home. You would have to get it disinfected or put in the autoclave before you can take it home. And the reason is we don't want students to take things into the lab which might pick up a microorganism and then students to take them home. So anything you bring into the lab which isn't brought into the lab, you put it in the cubbyhole and then you leave it there. Obviously, you yourself are walking into the lab, and anything on you or your person, like a watch, your glasses, a hearing aid, if you happen to have that, jewelry that you're wearing, can be brought into the lab, but you should not touch those items while you're in the lab. And that way microorganisms will not get on them and you will not take them home. Everything else should be left in the cubbyhole. And remember, the cubbyhole is outside the lab. And when you walk into the lab, you put your things in the cubbyhole, and that's not the lab proper. You then walk to the sink, and that's the lab proper. You then wash your hands, go put on your lab coat, go put on gloves, and then go to your lab bench. Okay. And then when you leave for the day, well, actually, let's start and do one more thing. Go to your lab bench and then disinfect it. Clear the area you're going to be working with of any microbes. And then do your work. And then when you're all done, before you leave, you disinfect your lab bench area again with disinfectant. And then you go and take off your gloves, take off your lab coat, walk to the sink, wash your hands, and then go to the cubby hole, pick up your things, and then you can leave. Any questions about any of that? That's the proper procedure for working in a microbiology lab, at least at Clark College. You come into the lab, and you're not really in the lab proper. You're just to the door in the cubby hole. Put your stuff in the cubby hole, and that way bacteria won't get on it. Your books should go there, your coats, your purse, your book bag. Anything else you don't want in the lab goes in the cubby hole. And then go wash your hands, go put on the lab coat, put on gloves, go to your lab bench, disinfect your lab bench, do your work. When you're all done, disinfect your lab bench. That's to kill anything that may have spilled on your working area, so the next person coming along won't get contaminated. Go take off your gloves, go take off your lab coat, go wash your hands, and then you can go and pick up your things in the cubby hole and then leave. Any question about that? All right. Only enrolled microbiology students are allowed into the lab unless authorized by the instructor. Yep, went too far, move that over. Paper and writing implements will be provided for you at your lab station. If you want to take notes, you can get a digital photo. Take the, what do they call that? An iPad, and take the photograph, email it to yourself, and that way you won't get anything from the notes or a picture or something you photographed. Cell phone usage in the lab is prohibited. If you get a call, you would have to take off your gloves, take off your lab coat, go wash your hands, go get the call, take the phone outside of the lab, answer the phone, or most likely recall them. When you're done, put away your phone, come back into the lab, put away your phone, go to the sink, wash your hands, put on your lab coat, put on gloves, and return to your workstation. If you were to use the phone in the lab, it would be confiscated and you would not get it back until it would be disinfected, either in bleach or autoclaving, and that would destroy any electronic equipment, like your phone. Anything taken into the lab that shouldn't be there would be confiscated for safety reasons. It would not be returned until it's been autoclaved or disinfected in something like bleach. Like I said, you are allowed to take some personal items into the lab if you're wearing them, like a jewelry, a watch, glasses. All cultures you will be working with are considered to be potentially pathogenic, so keep hands, pencils, and other materials out of your mouth, ears, nose. No eating or drinking, smoking, or gum chewing, or any other oral habit is allowed in the lab. You must always wear gloves, which would be supplied to you in the lab. You should, if you're working in the lab, monitor yourself for possible infections like diarrhea, fever, abdominal cramps. Obviously, we're going to be doing everything online, so that won't be a problem for you. And then we have a document which is on the online lab class. Information about bacteria used in the class, information for students and physicians, and you can find it on the class website. All students who come into the lab must wear closed-toed shoes. Any ladies' fancy open-toed shoes would not be allowed. Thongs on your feet or sandals are also not allowed. Tennis shoes provide little safety, but they do provide some safety. And so your shoes should be closed-toed. And the main problem we're concerned about is a spilling of microorganisms, like in a liquid culture, and it then gets on your feet. We don't want that to happen. The lab coats must be worn and completely buttoned when you're in the lab. Lab coats are supplied by the lab, but you may purchase a lab and then bring it to the lab. And then on the first day you bring it there, it must stay in the lab all term long. And then at the end of the lab, you will set it aside and put your name on it. And it will be autoclaved and then washed. And then it will be set aside for you to come and pick up after the term begins or after the term ends. Whenever you use something that's a splash hazard, that's a liquid culture containing bacteria or a dye that can do damage to the eyes, you must wear goggles. And goggles are supplied by the lab. They're kept in a box which uses ultraviolet light to sterilize them. Failure to wear goggles can lead to point loss. Obviously, once again, we're not going to be in the lab proper, so this is just an item you should know. The goggles must be worn when you're working with a splash hazard. Cleanliness is a virtual in microbiology, so you must disinfect your lab bench before beginning any work and after you're done with your laboratory work. And then you should also wash your hands before and after you do your lab work. You should note where fire extinguishers, fire blanket, eye wash station, and first aid materials are located. We will just talk about them in this coming lab. Obviously, we won't be in the lab, so you don't know need to know where they are. Any accidents should be reported to your instructor. Minor accidents should be cleaned up as follows. Always clean broken glass first. Broken glass can cut your skin, and the glass is likely to be coated with numerous microorganisms. So always use a broom and brush and a dustpan to pick up broken glass. Never use your hands. Dispose of broken glass in the broken glass receptacle. We put broken glass in a separate location in the lab, and it just is a container that says broken glass. Spilled strong acids and bases should be diluted and neutralized, and then wiped up. We don't really use strong acids and bases in the lab. Spilled bacterial culture should be cleaned as follows. You cover the spilled material with lots of paper towels. You then drench the paper towels with a disinfectant. You let the disinfectant stand for 15 minutes before wiping it up. You then wipe it up with paper towels. If there's broken glass in it, you take the broken glass out and put it in with the broken glass container. All the other stuff should be discarded in the biohazard trash can, which is I also called an autoclave container, because the biohazardous stuff is put in there and it will be autoclaved before it is put into the regular trash. Any question about that? So in the biohazard container should never contain glass. Glass should either go in the broken glass container or a a place to put glass, like glass pipette should be in the glass pipette reused area. Glass test tube should be in the reused glass test tube rack, discard place. Be sure to keep hair and paper away from Bunsen burners or back to incinerators. Long hair should be tied back, and it should be tied either before you enter the lab or immediately, well, before you leave your cubbyhole. Okay, so that's before you enter the lab proper. Your hair should be tied. Whenever you're not using the gas incinerator or the back to incinerators, they should be turned off. And they should be turned off before you leave the lab. Mouth pipetting is not allowed. Never take cultures out of the labs. Samples may be brought into the lab only when it's authorized by the instructor. If you have a cut or sore on your hand, you should wash your hands normally and then go to the first aid kit and put on a Band-Aid and then put the glove over the Band-Aid. As always, when you're done at the end of the day, you should wash or take off your gloves, wash your hands, and if you want, you may put on a fresh Band-Aid. Old specimens should be discarded properly. Agar plates will be a plastic plate, and they should be discarded in the biohazard container. Any material that's paper or plastic that has bacterial contaminants in it should also go in the biohazard container. Glass slides should not ever, and no glass should ever go in the biohazard container. Glass slides and cotton swabs should be put in a container for these items specifically, meaning we have a glass slide container that's filled with disinfectant, put your glass slide in that, and the swab, put it in the swab container, which also has disinfectant in it. And you can find those at the rear of the lab. Pipettes should be placed in the appropriate labeled container. If they're glass, put it in the appropriate pipette used container. If they're plastic and they're contaminated, they can go in the biohazard container. If they're not contaminated, like all you did was maybe pipette water, you can put the plastic pipette in the trash can. Glass test tubes should have their label removed and then should be placed in the test tube rack in the discard bin, and that's at the rear of the lab. Supplies needed for the lab will be at the front desk of the lab. Each pair of students are assigned a drawer and a lab bench, and so they share it. And remember, the area where the cubby holes is and your books and the door are considered outside the lab. All right. Students are required to follow these rules, and I'm just going to end it there. Any question about the lab safety rules? All right, if not, let's go on to Lab 00, lab safety training. Anyone working in an unsafe manner in the lab can be banned from working in the lab. Online students will not be working in the lab, but you do need to know this material, as many of these rules will be relevant during your professional career. The learning objectives, be able to, 1, distinguish between safe and unsafe laboratory practices, 2, understand where relevant safety equipment is located in the lab, 3, understand how to safely dispose of contaminated material. You are expected to observe a number of safety rules while you're working with pathogenic microorganisms in a microbiology lab. You're not actually going to be performing any of these because we're not going to go into the lab, but it is important for you to understand the guidelines which are required to create a safe laboratory environment. Safety includes wearing proper attire, such as a clean lab coat, disposable gloves, closed-toed shoes, and goggles when you're working with a splash hazard. These items are to remain in the lab for the duration of the quarter. And so you pick them up each time you come into the lab. The exception would be if you bring in a lab coat for the very first day. You only do that on your first day. And then it stays in the lab until after the term ends. Hands must be washed thoroughly with soap and water before you put on the gloves. Long hair should be tied back. No eating food or drink. No personal items allowed into the lab unless you're wearing it like a watch or glasses. The stuff that's kept in the cubbyhole, remember, we consider that outside the lab, and all personal items should be in the cubbyholes. Anything that's brought into the lab will be confiscated, and you will not get it back until it's been soaked in disinfectant for 15 minutes or put into the autoclave. If you come into the lab, you should be looking something like that: closed-toed shoes, long pants. You can't see it, but she's wearing a skirt or a blouse, a lab coat, gloves. I think those are her personal glasses. I don't think those are goggles. And she has her hair tied back. Or she's wearing a safety net, one of the two, which is the same. No food or drinks, no cell phones, no headphones. You always wash the area you're about to work with at the start of the lab, and then before you leave, you wipe it down with disinfectant again. And what you do is you spray it and then wipe it around, and then you actually allow it to air dry. We'll talk about that in the lab. This is an actual picture of our lab, and somebody would be working right here. And somebody would probably be working right there, but that could be a sink. I don't know. Can't see enough. All right. Discarding of the waste in the appropriate place is important. The regular trash, no sharp objects, no glass, nothing that's contaminated, like paper towels that do not have bacteria on it, like you use them to disinfect your lab bench at the start. You put it in the regular trash. Your gloves, if you don't get bacteria on them, they can go into the regular trash. If, on the other hand, you have bacteria on them, like you have a spill and you use the paper towel to wipe up the bacteria in the spill, or you spill something on your gloves, then take off your gloves and put it in the biohazard container, or wipe up the culture with microbes and put the paper towel in the biohazard container. We do have I already talked about that. No sharp objects, no glass in the biohazard container. Contaminated test tubes, like tubes that you're done working with, take off the label and put them in the test tube bin for old test tubes, which is at the back of the lab. There's a different bin for different items. There's a bin for glass pipettes, a bin for glass test tubes. We don't really use beakers, but you'd put that back there too. For slides, put it in the appropriate container. There will be a container labeled slides and has bleach in it. Put the slide in there. The swab, the cotton swab, put that in the cotton swab container. Once again, it has bleach in it. The cover slips, we have a container for cover slips. We do not reuse cover slips. Like the slides, they're put in the disinfectant they will be then washed and then dried and then reused. The cover slips we do not reuse and just put those in the cover slip container. You can disinfect it before putting in the disinfectant container. Sometimes the cover slip container will have disinfectant in it already, but they will not be reused. Consider everything you handle a pathogenic organism. If you're unsure of how to dispose of something, please ask your instructor. I already talked about spills, so I won't talk about that again. Lids should be on reagent bottles when not in use. Do not pour chemicals down the drain without permission. No mouth pipetting. Don't put anything in your mouth like your fingers, your nails, or a pencil. When a Bunsen burner or a Bacto incinerator are not being used, they should be turned off. A Bacto incinerator is a piece of stone that becomes red hot, and you put a tool in there to become red hot to sterilize it, and then you can use it after it's sterile. We use a Bacto incinerator. We don't use what are those called? The Bunsen burners anymore. Not unless it's an emergency. You should know where safety equipment is. Know that safety equipment is a piece of equipment that is there for your use to keep you safe. It is not something you use normally in the lab. Like the fire blanket is a piece of safety equipment, the fire alarm, the first aid kit right there. I think that's the fire blanket right there. The spill control station, an eye wash station, a fire extinguisher, a deluge shower. Those are all safety equipment. When in doubt, always ask. All right. So the lab procedures and safety rules. Never take corners and shortcuts. Always do the procedure that you're instructed to do, meaning and how you're instructed to do it. All right, this is the rules again. I'm not going to go over that. Accidents, I won't go over that. Never take cultures out of the lab. Okay, here's one. All microbial cultures must be labeled completely with your name, the date, the nature of the specimen. Like if you're growing E. coli, you just put E. coli. But if you're, like sampling the sink, say something like sink sample, the medium used. So you must include your name, the date, the nature of the specimen, and the medium used. It is optional. You may include the temperature you're growing the microbes at. I already talked about discarding things. I already talked about supplies. All right. I think that's everything. Any questions about this lab? If not, let's go to the worksheet. The questions don't show very easily with the lab module 00. This must be submitted by 8 a.m. Friday. this week, week 1, or you will be dropped from the class. So because the first questions didn't show, you can ignore the first page, and I have the safety word scramble here. Answer rule 1. In the lab, what do you always wear for safety before you enter the lab? And you must include 4 items. That's why there's 4 asterisks there. That is not really discussed in the lab module. However, you can see from the picture what she's wearing, what the 4 items can be. 2 of them you can actually see down here. And then one of them I mentioned, she's obviously wearing a shirt or a blouse. Okay. All right. Any question about any of that? So what do you put on before you enter the lab? And then rule 2, after you enter the lab, what do you put on to keep yourself safe in the lab? So this would be something you walk into the lab, go wash your hands, and then what do you put on to keep yourself safe while you're in the lab? These should be different than rule 1, by the way. Rule 2 and rule 1 should be different. Rule 3, this one you need to look up online. What is a safety data sheet? And each chemical in the lab has a safety data sheet. You need to describe what the safety data sheet does describe. I'm going to tell you a simple 1-word answer will not be enough to answer the rule 3. You need at least a phrase. Okay. Rule 4, know the location of your safety equipment, such as, and then you have to answer 4 different things. Excuse me, 5 different things. Okay. And don't bother erasing the asterisks. I know how many asterisks are there, and so you can't short it. Rule 5, what can you take into the lab? That could be 1 or 2 things. And then can you eat or drink in the lab? And that's either yes or no for the third item. And then 2, list 1 item of laboratory attire that you must wear in the microbiology lab. 3, list 1 item that is allowed in a microbiology lab. And then 4, list 1 item that's not allowed in the microbiology lab. And then 5, make sure you submit this worksheet by 8 a.m. Friday of week 1 in the folder to worksheet lab 00, or you will be dropped from the class. I will use this as attendance to determine who is showing up for our class. So make sure you get this assignment in by 8 a.m. Friday. All right, so go ahead and work on the lab. I will be here until 8 o'clock to assist you with any questions. After 8 o'clock or after the last student logs out, I'm going to log out. So if you have questions, make sure you ask it early before 8 o'clock or after the last student logs out. Any questions? All right, go ahead and get started on the lab. I am here to help you.